In this video we will discuss about the pancreatic cancer cells. So the area that we'll cover about the pancreatic cancer cells is its property uh, to be resistant against chemotherapy and the radiotherapy. So we will start with these stellate cells. The stellate cells in the pancreas helps in the formation of ECM, that is extracellular matrix that surround the pancreatic cells. However, when the pancreatic cancer cells are present, it will try to induce or try to activate the pancreatic stellate cells to form fibrotic uh, more fibrotic like ECM. And because of this, what will happen is that it will induce the hypoxia conditions, that is amount of oxygens that are required for the normal functioning of the cells is not available within the pancreas, and this hypoxia condition will further stimulate the more fibrotic like ECM formation. So this pancreatic cancer cells will leads to the generation of the perpetual motion of the hypoxia induced fibrosis, and this hypoxia-induced fibrosis act as a physical barrier that will prevent the chemotherapy, the anti-cancer agents that are available in the chemotherapy and the radiotherapy to uh, affect these pancreatic cancer cells. And apart from this fibrotic ECM, the other thing that desmoplasia reactions that will also act as a physical barrier that will not only prevent the chemotherapy and chemotherapeutic and radiotherapeutic agent to act against the pancreatic cancer, but it will also prevent the immune cells to come and interact or trigger the immune response against the pancreatic cancer cells by acting as a physical barrier. Apart from that, this ECM will also induce this EMT characteristic, that is epithelial to mesenchymal transition. We have discussed about this, what does this EMT means in our previous videos. So what happens in brief is that the pancreatic cancer cells, which are by nature has a epithelial characteristic that they will remain in their that is they don't have the ability to move from one place to another place, will acquire the mesenchymal characteristics that will help the pancreatic cancer cells to move from the uh, site of origin to the secondary site where they will infect the secondary organs, and this can be done with the help of blood vessels, and we have discussed that thing in our videos about how this extravasation intravasation could lead to the pancreatic cancer cells to spread across different part of the body, such as lymph node, liver, etc. And apart from these factors, the other factors that also helps in the chemotherapy and radiotherapy resistance is the reprogramming that happened within the pancreatic cancer cells. So even though some of the chemotherapeutic agent able to enter inside the pancreatic cancer cells, the pathway that these agents are targeting are already been reprogrammed in the pancreatic cancer cells. For example, the apoptotic pathways are already been reprogrammed that have been targeted by some of the chemotherapeutic and radiotherapeutic agent. Thereby, this anti-cancer drugs will have very minimum impact on the pancreatic cancer cells. At the same time, the reprogramming also ensure the survival and proliferations within the pancreatic cancer cells, and thereby the upregulations of the survival pathways ensures the minimization of the impact of this chemotherapeutic and radiotherapeutic agent. For example, the p53 pathways has been p53 gene has been downregulated, whose function is to trigger apoptosis whenever the damages are beyond repair within the DNA. So this in this function of this pancreatic cancer cells resistance, these surrounding cells such as activated pancreatic stellate cells also plays a important role to ensure a physical barrier to these pancreatic cancer cells, and thereby helping those helping them in helping them in towards helping them from these anti-cancer drugs And at the and apart from this combination of the combination of these stellate cells and this ECM, and along with these cytokines that are released by pancreatic cancer cells, will form a tumor microenvironment which will have a uh, reduced pH or a changed pH, and this pH will also helps in reducing the efficiency of the anti-cancer drugs and radiotherapeutic drugs, as they will be less effective in those pH that are present within the tumor microenvironment. So this is some of the additional ways by which pancreatic cancer cells works against the chemotherapy and radiotherapy. If you want to understand more about these pancreatic cancer cells, uh, functions, or how they avoid or dodge this chemotherapy and radiotherapy, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.